Okay, good afternoon. So this is a webinar by our visiting fellow, our visiting MASTS fellow, Professor Jonathan Erez. Jonathan is at the Institute of Earth Sciences at the Hebrew University of Jerusalem in Israel. He's worked extensively on biomineralization of marine calcifying organisms, particularly forams, corals and mollusks. And the title of his webinar today is Biomineralization Mechanisms and Proxy Incorporation in Marine Calcifying Organisms. There will be time for questions and answers at the end, but Jonathan will start his webinar now.
Thank you, and I thank MASTS for the opportunity to come and speak here at St. Andrews University. And what I'm going to show you today is mainly it's mainly things that are related to biomineralization in foraminifera, which are considered the best proxies for paleoceanography. But as usual, I'm only giving the talk, but there are many participants in these studies, and they are named here. The first line is really the graduate students that are involved in this work. The second group is colleagues, and the third group is people who helped us technically. And I'm going to start by talking on the importance of calcification in the ocean as part of the global carbon cycle. I will then talk about the effect of ocean acidification on marine calcifiers. And then I will talk a little bit about proxies in paleoceanography and why they are important. And then I will tie both subjects, the ocean acidification and the proxies, into the biomineralization mechanisms of these organisms. And basically, we will try to resolve this issue using experimental work that we have done with fluorescent dyes. And the next one will be, and that is the main message of this talk, that marine calcifiers bring seawater to the site of biomineralization. And then it will be possible to explain the sensitivity to ocean acidification and the successful use of proxies. So I will start by showing you the famous figure that everybody shows in such cases, the increase in atmospheric CO2. We are now above 405, and this is an unprecedented increase in atmospheric CO2 concentrations. The truth of the matter is that the emissions to the atmosphere is shown by the upper line, and can I show it like that? Yeah, in the upper line, and that is what it should have been, but in reality, in the atmosphere, we measure the lower lines, and that is because good part of that CO2 was already absorbed by the ocean and by the terrestrial ecosystem. So the oceans takes about 50% of everything that was emitted to the atmosphere, and therefore it becomes acidic. And just to show you how dramatic is this increase, here we have a record of CO2 in the atmosphere that was taken from ice cores and from from trees, and you can see that it was around 280 for about 1,000 years, and since the Industrial Revolution it started to go up dramatically, as you can see here. And this is an unprecedented concentration and also extremely high rate. Now, if you look at the global carbon cycle, then you can see that the atmosphere has a very small reservoir of only several hundred gigatons, and the ocean, on the other hand, the upper ocean has about the same amount of carbon as inorganic carbon, but the deep ocean has about 40,000 gigatons of carbon, and obviously the ocean is controlling the entire global carbon cycle. And in the ocean, there are two processes that really control the carbon. One is photosynthesis, that is shown here in the upper ocean. CO2 is taken up and producing organic matter and oxygen. And the other process, which I'm going to concentrate on today, is calcium carbonate precipitation, which is shown by the second reaction here. Calcium carbonate is precipitated by organisms, and these two processes control in addition to, of course, inorganic chemistry, they control to a large extent the atmospheric CO2. And if we are to understand what is going to happen in the future, we really need to appreciate these processes and describe them in quantitative way. In the deep ocean, by the way, you can see oxidation of organic matter and calcium carbonate dissolution, which are, of course, the opposite reactions that we see in the surface ocean. Now, marine organisms are the ones who are calcifying in the ocean. And if you look at sedimentary rocks like chalks and other limestones, if you look inside, you can see that they are really made of foraminifera and coccolithophores, which I'm going to expand on in a minute. And here are, for example, the coccolithophores. These are very small marine algae that photosynthesize, but in addition to the photosynthetic activity, they also build these beautiful coccoliths, which are made of calcium carbonate. In the sediments, they look like that. Of course, it's a mixture of many species, and the coccosphere, which is the cell, is now separated into the coccolith that came off this shell. And the foraminifera, as you can see here, this is from the Gulf of Eilat. These are planktonic foraminifera. They are very big. You know, the coccoliths are maybe 10, 20 microns. The foraminifera are about 1,000 microns. So they are about 1 millimeter in size. They are very big and they are very abundant. What you see here is not something that we picked to show you the foraminifera. It is just normal marine sediment that we filtered through about 150 microns, and that is what is remaining. So you can see that this is a very important component of marine sediments. And in the past, we know that not only planktonic foraminifera were so abundant, but also benthic foraminifera. Here, for example, you see a picture of the nummulites. They were huge unicellular benthic foraminifera. They really thrived in the Eocene, and these pictures are from Egypt. And you can see how big they are. They were called nummulites because they remind you of money. And here you see these pictures are actually taken from the stones of the pyramids in Egypt. So the entire pyramids are built from these nummulites. So it's not the children of Israel who built the pyramids, it's really the nummulites. And here you can see another important component of the global carbon cycle in terms of calcium carbonate precipitation, and these are coral reefs. This entire structure that you see here is a coral reef that was built by corals, and they are really important component of the whole system. Here you can see the corals themselves that build the coral reefs, and I'm not going to expand on corals today. I just bring them for completion of the picture, and I will talk a little bit about how they respond. Now, the main talk is going to be on foraminifera. So here you see a live planktonic foraminifera. It looks quite different from the ones you saw in the sediments because it has long spines and it has those little dots, which are symbiotic algae that photosynthesize while this organism is calcifying. And recent budget that was done on calcium carbonate production in the ocean shows that foraminifera produce about 50% of the calcium carbonate, coccolithophores about 25%, corals about 12 or 13%, and echinoderms of all things are also very important in this budget. The total precipitation of calcium carbonate in the ocean is about 1 gigaton, compared to what is being emitted to the atmosphere today, which is 10 gigaton of carbon. So this is really, it looks like a small amount, but before humans started to interfere with the ecosystem, that was a very important component of the system. Most of it is, by the way, dissolving. 80% of it is dissolving, and only 0.2, 20% of that is accumulating on the ocean floor. Okay. In about 2000, this paper by Ribesal et al. Came up, and they showed that coccolithophores, when are exposed to higher CO2 levels, that you can see here, are calcifying less. You can see the rate of calcification relative to the organic matter production decreased in 2 species, and that paper started the whole wave of studies where people were trying to look at the effect of ocean acidification on calcifying organisms in the ocean. The coccolith story is not so simple, because soon after that paper, there came several reports that actually showed the opposite, that coccolithophores are not really suffering from ocean acidification, but on the contrary, they tend to calcify more. And this record by Iglesias Rodriguez, as you can see, they show that at low CO2, they have small coccolith, and at high CO2, they have big coccolith. And you can see that the volume of the coccoliths have increased with CO2. In a way, this is not surprising, because coccolithophores are algae. If you give algae more CO2, they grow better. But the question is why they increase their calcification as well. And I want, before we continue, let us look a little bit at the carbonate chemistry of the oceans in order to understand what is the effect of ocean acidification. So the total inorganic carbon in the ocean, that 40,000 gigatons that I talked to you about before, is composed of 3 species: CO2, bicarbonate, and carbonate. And the pH and it's a function of pH. The pH of the ocean is around 8, so it's marked here, seawater, and you can see that bicarbonate is the dominant one. We have a little bit carbonate ion concentration, and the CO2 is very small. If you have ocean acidification, we reduce the pH. And then what happens is that bicarbonate goes up, and the carbonate ion concentration really goes down. And that's really important, because the calcifying organisms are dependent on the carbonate ion concentration. And this I'll show you in a minute. So the net effect of ocean acidification is to increase a little bit the bicarbonate, but decrease the carbonate ion concentration, and also increase a little bit the CO2. Okay, so if we go to corals, for example, this is an old experiment that we've done in 2008, Kenny Schneider's PhD work, and you can see here the calcification rate of a coral as a function of pH, and you can see that at higher pH they calcify a lot, at lower pH they calcify less. The difference between the 2 lines is that this is calcification in the light, and this is calcification in the dark. Now, this very same data set can be presented not as a function of pH, but as a function of the carbonate ion concentration, and the picture is the same. So we don't know if this is now a pH effect or the carbonate ion effect. And to resolve that issue, we ran another experiment in constant pH, and another experiment in constant PCO2, and you can see that we see the same relationship if we plot the calcification rate relative to the carbonate ion concentration. So remember, this is done now in constant pH. We only change the total CO2, and as you can see here, they show exactly the same trends. So based on this study, we concluded that the carbonate ion concentration is the one which controls the calcification rate in corals. Now, the same thing can be done on many other organisms and have been done by many other researchers on other organisms. Here is our small contribution here with relation to foraminifera. You can see them here. And here is the calcification rate of Operculina ammonoides. This is a big foraminiferan. Although it's unicellular organism, you can see it reaches some 2 or 3 millimeters in size. And you can see here the pH. And there is this clear relationship that when the pH is reduced, the calcification rate is reduced all the way to dissolution. Okay? So next, we're going to go into the entire coral reef because instead of studying every organism that lives on the coral reefs, and the diversity is huge, we decided to go and do community metabolism for the entire coral reef. In the case of the coral reef in the Gulf of Eilat, in the very northern tip of the Red Sea, it is really easily accessible, and this is the coral reef. This is the underwater observatory of Eilat, and here is the site of our study. So we are basically monitoring the chemistry of the seawater as they flow over the coral reef and out of the coral reef. And when they are over the coral reef, they accumulate some signals from the metabolism of the coral reefs, and we can calculate, based on that, the community metabolism. When we do that, so this is the calcification rate in millimoles per meter square per day of the coral reef. And this is the omega, which is defined as the calcium times the carbonate ion concentration divided by the solubility product of calcium carbonate. But because calcium and K almost do not change at all, carbonate is really controlling this equation. So omega is controlled by the carbonate ion concentration, and as you can see, this is really the main line we have to concentrate on. There is a huge difference between calcification when omega is 4.3, and it is really reduced dramatically by a factor of maybe 5 or more when it is lowered only to 3.7. We are still supersaturated, but there is a very strong sensitivity for the entire reef community, corals, algae, mollusks, foraminifera, and many others, to the carbonate chemistry of the water. Now, this is a little bit misleading because during these changes, which are basically, if I go back for a minute, are changing between the summer here and the winter here, we also have a change in temperature. So in order to take care of that, we corrected for the effect of temperature, which is shown in these lines. So this is the dependence of calcification on omega at different temperatures. These are the dashed lines. And our data are plotted here. So you can see that this is really a combination of both temperature and omega. And if we then clear off the effect of temperature, we remain with the net effect on omega only. And to test whether this is really applicable to a serious and big coral reefs, we went to the Great Barrier Reef in Australia. It's 2,000 kilometers almost in size. You can see it from satellites, as you can see here. And we went to 2 locations. One is Lizard Island in the north, and the other one is One Tree Island in the south. And here you can see, so these are beautiful coral reefs. They are still maintaining their very healthy look, and they are calcifying, and they are in good shape. And the main reason we went there was really that the same coral reef has been studied many years earlier by other team of scientists led by Dave Barnes and his colleagues, and they were doing very similar measurements of community metabolism. So that was in the 1970s, and we decided to go there so many years later, in the 1990s, at the end of almost 2000, and see what happened between those periods, because since then, the CO2 have increased a lot. So this is how much this coral reef calcified in 1975. And this is how much it calcified when we came to Lizard Island in 2008. You can easily see a reduction of about 40%. And it was so amazing that we couldn't believe it. We went again in 2009, and we were able to measure similar rates as in 2008. And you can see that the CO2 in the atmosphere in 1975 was 330 parts per million, and in 2009 it's 390 parts per million. So 60 parts per million were enough to lower the calcification rate of that reef by 40%. And how do we know that it is caused only by CO2? Because these are the relationships that we developed from the coral reef in Eilat and other coral reefs in the world, showing the dependence of calcification rate on omega, on the carbonate ion concentration. And as you can see here, Lizard Island was here during the 1970s, and it is now here during 2008 and 9. So it falls more or less on the line as expected, and therefore we believe that the main cause for this decrease that you saw here is really the ocean acidification. So, short summary, many marine calcifiers include coral reefs communities decrease their calcification rates in response to ocean acidification. And they respond to omega, or to the carbonate ion concentration, as if they are composed of inorganic calcium carbonate, because the experiments that I showed you before were done on inorganic calcium carbonates, and that is exactly what we would expect. And this is strange. Why is it strange? Because marine organisms are not supposed to respond as if they are inorganic calcium carbonate. Marine organisms are protected from the environment. They have their tissues, they have their cells, they are protected by membranes and epithelia. And why, therefore, they are responding in such a way to the ocean acidification? That is a valid question. And indeed, the coccolithophores, which are well protected from ocean acidification, and I'll show you in a minute why, are not so sensitive to ocean acidification. In fact, they show sometimes increase and sometimes decrease to the ocean acidification effect. So now the common biological thinking about biomineralization is that the organism creates a privileged space where the mineral is being deposited, and that ions are supplied to that site either by membranal transporters, which are usually proteins that can be either channels or pumps, and the chemical microenvironment where the biomineralization takes place is really controlled by the organism. It has its own pH and ionic composition and supersaturation and whatever. And there is also organics involved. Lots of the crystallography and the mineral structure are controlled by organics that the organism is precipitating, is excreting into that calcification space. And coccolithophores are indeed the classical example for it. So let's look at the coccolithophores. Here are the calcium carbonate protective shells that you can see here. And the calcium carbonate is really produced inside a big vacuole that is inside the cell. So we have the cell membrane and we have this vacuole membrane. Both of them are separating the site of coccolith formation from the seawater, which is outside. Okay, here you see it in a TEM picture, and here you see the schematic of it. Okay, so here's a model that people put to explain what is happening, for example, to the calcium. Calcium have to enter the cell through the membrane, through a transporter, so a calcium channel, and then it comes to the cytosol. From the cytosol, it needs to be transported again by another transported into the coccolith vesicle. So in order to arrive to the site of biomineralization, the calcium ion and the carbonate as well have to pass through two membranes which are completely separated from the bulk seawater on the outside. If this is the case, and this is really the true mechanism, then why should they be sensitive to a little drop in pH, which is 0.1 or 0.2 or 0.3 pH units? Okay, and indeed, they are not so sensitive. If we apply the same thinking, and this is what was the common thinking about coral calcification, still is, I think, today. This is summarized by Aleman from Monaco, and they show calcium coming into the here's the calcium carbonate skeleton, here's the calcifying fluid. Calcium is entering through a channel, and then it is pumped by a protein which is called calcium ATPase, requires energy, transports the calcium across the membrane again into the calcification site. If this is the true explanation of how corals calcify, so why are they sensitive to ocean acidification? You know, it is really everything is happening here in the cell. Why should they worry about if out there about another 2 layers of cell, the pH is dropping by a small amount? Well, we work on foraminifera mainly, and in the end of the 1980s, we published a bunch of papers with Benno Terkaule and myself, looking at the calcification mechanisms of foraminifera. We adapted the regular biological explanation. Here is bicarbonate being pumped in, protons being pumped out perhaps, calcium is being pumped into the cell, and it calcifies. Well, because it's our work, I can really say it is really wrong. The new observations show us that we could not accept that model, not for foraminifera, not for corals, maybe yes for coccolithophores, but not for the other organisms. And you see, the geochemists, on the other hand, were always considering foraminifera and corals and mollusks and other calcifying organisms as little pieces of calcium carbonate that float in the ocean and behave like inorganic. And so that is why they were using proxies to determine the paleoceanographic conditions. And they say, yes, there are some vital effects, but by and large, the calcification of organisms is very similar to inorganic precipitation from seawater. And they were hoping that vital effects, which are those small deviations, will remain constant and will not jeopardize our ability to do paleoceanographic work. And in that case, we ask really why all these paleoceanographic proxies that you see here are working. Because if indeed the mechanism is similar to what coccolithophore people tell us, then the calcium carbonate should be so well protected from the marine environment that it should not record all these trace elements and stable isotopes that you see here, which are the paleoceanographic proxies that are being used today. They should not be able to really record them faithfully and so that we can reconstruct the past ocean conditions based on these proxies. But the fact is that indeed they record these proxies beautifully, and here you see a long history of oxygen isotopes in benthic foraminifera. This is going back 70 million years, and you can see a beautiful record that shows you that by and large we started in the Eocene. It was very warm temperatures. It will probably, so this is the deep ocean picture because it's based on benthic foraminifera that live on the ocean floor. And you can see that the temperatures that today in the ocean floor are around maybe 0. At that time was something around 12 degrees Celsius. And it has been cooling, especially during the Eocene-Oligocene transition, and then it has been cooling ever since towards the Pleistocene, and all this big noise that you see here are the glacials and interglacial cycles that I cannot dwell on. And this record was somehow backed up by some by atmospheric CO2 estimates. And so the very few estimates that we have for CO2 that at that time in the Eocene, the CO2 levels were about 2,000 parts per million, whereas today we are talking about 300, 400 parts per million. So this is why we think that there is a strong sensitivity to our climate to CO2. And, you know, later on, the CO2 levels went down and the temperatures went down. Okay? So, now, when we think about the complexity of biomineralization, as I described it before, we ask ourselves, how come that all these paleoceanographic proxies are working? How does it work? They should have recorded whatever is happening in their own cells and not in the seawater. And just to show you that it really works, at the time that we published this work, there were really questions whether the delta O18 records properly the temperatures of the water. And this is the first paleo-temperature equation that was determined experimentally on planktonic foraminifera. And we cultured these delicate organisms in Eilat. And here is a culturing temperature. Here is a delta O18. And the line that goes between the points is not the best line. It is just the predicted line according to the paleo-temperature equation of Epstein et al. From the 1950s. So foraminifera are well behaved, even at a range of temperatures that is way beyond where they live. You know, here it was 28, and down here it's about 16. This is really at the lower edge, below the existing temperatures, and they were really well behaved. So we were really surprised by that. Now, so here are these planktonic foraminifera again, and before we continue, let me explain why everybody is working on planktonic foraminifera. That is because they were really intelligently designed for paleoceanography. And they are big. You can easily see that we have different species, and you can pick them one by one. And then you can take them for isotopic measurements. And they are highly abundant. They preserve well in the record because they are made of low magnesium calcite. And most important of all, and this is really one of the main messages of this talk, as I said already before, they precipitated their shell directly from the seawater, not at all like coccolithophores do it, okay? And the rest of my talk will be really to show you how they do this calcium carbonate precipitation directly from seawater, or almost directly from seawater. Okay, and how do we know that? Okay, the reason why we know it is because we have a direct proof that seawater arrives to the site of calcification, both in corals and in foraminifera and in mollusks and in sea urchins and in almost any other organism that we have touched. The only one that did not stand to that test are the coccolithophores. So instead of taking the coccolithophores as the model for biomineralization in the ocean, they are really the exception, okay? They are still having their well-respected position in the community. However, they are really exceptional, and all the others do it in a totally different way, not by pumping and channeling the ions into the site of biomineralization, but instead by bringing seawater to the site of biomineralization. Okay, so the practical proof that this is indeed the case is the fact that when you take these organisms and culture them in seawater and add a fluorescent dye to the seawater, in this case it's a dye called calcein, and it has a high molecular weight of 600. Here is a molecular structure of calcein. You can see that the skeletons get labeled beautifully with calcein. And these are planktonic foraminifera, these are benthic foraminifera, and you can see that the calcein is incorporated into the calcium carbonate, because what you see here is after we clean the shells with sodium hypochlorite, which removes all the external non-fixed calcein, okay? So what is so special about calcein? What is the significance that these dyes are incorporated into the shell? The significance is that both calcein and another dye that we have been using, FATC, connected to a large sugar, dextran, 10,000 usually, are large organic molecules that cannot penetrate through the membranes of a cell. Okay, so they are cell impermeant. And this is why the coccolithophores, for example, are not labeled with calcein, because calcein cannot go through the membrane of the coccolithophore. The ions, like calcium and probably bicarbonate, can go through the membrane, can be transported, but the calcein and the FATC dextran cannot penetrate through the cells. But it does arrive to the site of biomineralization in foraminifera and in corals and in sea urchins and in mollusks. And so the question is, how does it happen? And the important thing here to remember is the bare fact that these skeletons are labeled with a fluorescent dye is proving that seawater is getting there. So let me show you how it works. So here is our pet organism. It's Amphistygina lobifera. It's a big benthic foraminiferan. This is like 1.5 millimeters in size. And the only reason I show you that is because to show you that they build chambers one after the other. And the chambers of this species are lobate. This is why it is called lobifera. You can see here the structure and you can see these beautiful lobes. Okay. Now, due to very lucky experiment that we have done, we were trying to partially dissolve them, and then when they recalcified, instead of building their lobate chambers on themselves, on their previous shell, they precipitated their shell on the glass slide on which they were sitting. And here you see some of these lobes that they built as a new chamber. Now, these lobes are therefore allowing us to look with the inverted microscope and really look at the calcification process. So what you see here is before it starts to calcify, you can see here the pseudopods, you can see the cytoplasm, and you can see some symbiotic algae in here. And then the next stage, we can see already some calcium carbonate being precipitated on these lobes. And in the next step, a few hours later, you can see that the whole thing is full of calcium carbonate precipitates. And all that is still done underneath this layer of cytoplasm that the organism is creating. The model, I cannot show you the movie, but the model that shows this is seen here. So here is a foraminiferan with one chamber, and it has pseudopods. And what we think is happening, it's vacuolating the seawater. And vacuolization or pinocytosis or endocytosis is a well-known process in biology. This is how these organisms feed. For example, if there was a diatom here, they would engulf it like that and digest it, okay? So they know how to do that, but at the same time, they can just vacuolate seawater. And they also can vacuolate themselves like that. So then they isolate themselves from the seawater here, but still they are able to bring seawater to this site, either by transporting the vacuoles of seawater into here and exocytose them, or by even opening such openings that you can see here or here or here. And then they can precipitate a new chamber, as you can see here. And this is a privileged space that we talked about, but it's full of seawater. And it's full of seawater with the fluorescent dye. And therefore, the fluorescent dye gets incorporated into the skeleton. Okay? So here's just to show you how intensive is this seawater vacuolization process. You can see that almost the entire volume of live foraminifera is full of seawater vacuoles. And this is through the shell. So they are live specimens. And again, big vacuolization, high magnification. This is the external seawater. This is the internal part of the organism. In red, you can see the fluorescence of the symbiotic algae, and here you can see the seawater vacuoles, which contains a fluorescent dye. I cannot play the movie because it cannot be recorded, but let us go to the next step. In the next figure, you can see, again, seawater vacuoles of calcein inside such a foraminifera in high magnification. And during the labeling period, which was about, maybe 6 hours or something like that, some of the calcium carbonate that they precipitated got labeled with calcein, as you can see here. But then we took the organism, moved it to another dish of seawater without calcein, and in the next morning, when we started to look at it, it was heavily calcified with calcein, so much more fluorescence than in the previous day. And remember, the seawater had no calcein. So where did all this calcein came from? It must have come from the seawater vacuoles that were inside the organism. Okay, so this is the direct proof that the seawater vacuoles are serving for the calcification process in foraminifera. And later on, when we understood that this is what was going on, we started to use pH-sensitive dyes. This one is called SNARF, and you can see here the pH scale. And this is the seawater, this is the calcium carbonate of the organism, and this is the internal cytosol. And in the cytosol, you can see big vacuoles of seawater, and you can see that if you look at the color here and compare it to this scale, you can see that this is really higher pH than the seawater. And if you calculate the pH here, it ranges between 8.5 to about almost 9.5, and on the average, it's about 9. Okay, so the seawater vacuoles are then being modified by the organism, and the main modification is that it goes to higher pH. There are also acidic vesicles that are found here, and they may supply CO2 to go into the basic vacuoles. I don't have too much time to go over it. This is from a PhD thesis of one of my students, Nivi Kessler, and here you can see the distribution of pH in the seawater vacuoles, and you can see it here. So as I said, the average is around 9, but it can be higher and slightly lower. It is usually never lower than the seawater. Okay, good. So here's a schematic that explains what is happening. Here's a foraminiferan. It has these basic or alkaline vacuoles, as you can see here, with pH 9. It has these acidic vesicles of seawater with pH 6. CO2 is then released into the cytosol and is being absorbed by the high pH vacuoles because CO2 can diffuse freely through membranes, and therefore these are big sinks for CO2, and these foraminifera accumulate inorganic carbon in very large quantities, and this is something that we discovered many years earlier. I don't have time to talk about it, but it's all published with a bunch of papers around the 1990s with Benoter Kaule and others. And at that time, we did not understand why they have internal carbon pool. Now we can start to understand. If indeed the calcifying fluid here is seawater, seawater has a calcium concentration of about 10 millimolar. Okay, that's a very high concentration. The cytosol here has a calcium concentration of 0.1 micromolar. So this is like 4 or 5 orders of magnitude lower than the concentration in the seawater vacuole. Okay, now calcium is very plentiful in the seawater, but we don't have enough CO2. So the organism is accumulating the inorganic carbon in order to precipitate calcium carbonate. And how does it concentrate the inorganic carbon? We found just by, again, some luck, we think that it is mostly done by a sodium proton transporter that pumps sodium into the seawater and hydrogen ion out of the seawater, so that the pH is increasing. and then CO2 is diffusing inside. So how do we know that? Well, there are two indications. One is if we look at the boron isotopes, which is a very famous now proxy for pH in the ocean. So I'm not going to go over the details of this. This is well published by many others, but there are two species for the boric acid in seawater. There is the boric acid itself and the borate, and the abundance of them depends on the pH. So the pK here is about 8.6 or 8.7, something like that. And there is a big isotopic difference between the borate, which is here, and the boric acid. Okay, so if we change the pH, as you can see here, and we assume that borate is the species that is incorporated by the calcium carbonate skeleton, then if the pH is increasing, the delta boron 11, or the ratio of boron 11 to boron 10, is increasing, as you can see here in this line. Okay, so these are some previous data, and our data plotted like that. So this is really interesting because here is a delta boron 11 as a function of pH. This is experimental work, so we've controlled the pH really well. And these are the measurements that were done with Claire Rollion-Bard in Nancy in France. We're using the SIMS machine. And I'll switch to this figure, which translates the delta boron 11 into pH. So this is the calculated pH based on the boron isotopes, and this is the culture pH of the seawater. And the interesting thing you can see here that on each specimen we did quite a few measurements, and in addition, for each treatment we took several individuals. So we have statistics, and you can see them here, and there is a big variability in each treatment. And the surprising thing is that all of them show the highest pH around 9, okay? And the lower pHs are going all the way down to the ambient pH of the seawater, okay? So if we actually took the lowest values for each treatment, we will get the exact one-to-one relationship, and we could really take the pH measurements, okay, of the ocean or of the water. But in reality, what we see is a big range of pH. At the top of it is around 9, okay? That is exactly the pH that we saw in the seawater vacuoles, okay? And what is happening, we think, is that indeed they elevate the pH, they accumulate inorganic carbon, and then when they start to precipitate the calcium carbonate, the pH goes down, because this is what should happen when calcium carbonate precipitates, and it reaches almost the values of the ambient pH water. Okay, the second interesting proof for that mechanism comes really from lithium isotopes, and that is work that was done in the same lab by, again, Claire Rollion-Bard and Nathalie Vigier and myself and another student who cultured these foraminifera. And this is published in Comteren 2 Geosciences in 2015. And you can see here that if you measure the lithium isotopes as a function of temperature, no change. You measure the lithium isotopes as a function of pH, no change. So that was very boring. But then suddenly, we did an experiment where we kept the pH constant, you see here, about 8.16 at constant temperature, and then we changed the DIC. And when we change the DIC, suddenly we see a huge change in the lithium isotope composition. And these are all the previous experiments, you know, the ones that were in the different temperature and different pHs, and they show us, so they all fall on the same line. And that was really very, very surprising. And at the beginning, we couldn't believe that this is happening, because this is a huge amount of fractionation. It's like 20 per mil. And the only place where we find such fractionations in nature is really in photosynthesis, for example. Plants are fractionating carbon isotopes about 25 per mil or something like that. And this is what our foraminifera are doing in the calcium carbonate with lithium isotopes. So in addition to that, we also measure the lithium to calcium ratio. And if you plot the lithium to calcium ratio against the lithium isotopes, you can see that there is a very clear relationship here. So this is the experiment with the high DIC, above 2.5 or something like that. And the isotopic composition is heavy, and there is very little lithium in the shell. Okay, but if you go to the low DIC, here we have a lot of lithium in the shell. The lithium to calcium ratio is really increasing by a factor of more than 10, and the isotopic composition is very light, okay, as you can see here. So, and these are, by the way, the pH and temperature data, and these are previous data that other people published on lithium isotopes and are following also on our line, okay? So, how can we explain that? So the explanation is here. So it's the same model that you saw before. Here we have the basic vacuoles, the alkaline vacuoles. Here we have the acidic vesicles. I'm not going to talk about them, but look only here at the alkaline vacuoles, which are the raw material for the precipitation. So it's basically seawater, and now we want to elevate the pH. If we use, in order to elevate the pH, the sodium proton transporter, which requires energy, it requires ATP, then because it goes against the gradient, we pump sodium into the vacuole. Those of you who are familiar with carbonate chemistry, it's basically pumping in alkalinity, and we pump proton out. Therefore, the pH here is increasing, okay? The pH is increasing, but at the same time that sodium is being transported here, also lithium, which is having very similar chemical characteristic like sodium, and is well known in biology to replace sodium in such processes, is also entering the vacuole through the same transporter, but it's probably isotopically fractionated strongly, and therefore, if we have low DIC, so for example, if we have low DIC, we have low DIC values in the seawater, this pump has to work extra hours if you want, and therefore transport more sodium and more lithium into the vesicle, into the vacuole, and at the same time fractionate the lithium isotopes. And if this is true, then we can use the lithium calcium ratio and the lithium 7 to calculate the DIC, the total CO2 concentration in the past ocean, and that is of course extremely important because together with boron isotopes, we can now determine the entire carbonate system, and then we can predict what was the CO2 levels in the past oceans. So this is really what I wanted to show in this short presentation. So to summarize, many marine calcifiers reduce their calcification rate in response to ocean acidification. This is because they bring acidified seawater to the site of biomineralization, and they are open to the ocean, so to speak, and this is why they are so strongly affected by the ocean acidification. This novel pathway for supplying, this is really a novel pathway for ion supply for marine calcification that was not considered before. This mechanism explains why we can use foraminifera and corals and mollusks and probably other organisms as such good proxies for paleoceanography. And it is also explaining us the ocean acidification, as I said. So this is all, and I want to thank you.
Thank you very, very much, Jonathan. I don't know if there are any questions, but if there are, could you please enter them into the webinar software?
Okay.
I think we'll finish there.